And share. There we are. All right, so here we are. Boom, boom. We're here. All right, so there's a quick video to show you how that looks. All right, so watch closely. It's very short.
The individual characteristics of each one of us. When the two homologous chromosomes lie side by side, breaks occur in the chromosomes and corresponding lengths of the chromosome are interchanged. This crossing over disrupts the original sequence of the alleles and interchanges parts of both maternal and paternal chromosomes so that the possible number of gametes with different genetic information that any individual can
Oops, sorry.
produce is almost incalculable. This is why a child may inherit his father's nose and his mother's eyes.
All right, so you get this ability for genes to hop. Now, one thing I want you guys to remember, so when you took your stuff home to make your dragons, you took 2 letters for each trait, right? And those are alleles. All right, and alleles are part of a gene. There are multiple allele. How do I put this? Not alleles. Actually, scratch that. I'm going to come back to it because it can get a little confusing because we're talking about when we start talking about DNA and nucleotide bases. And then there are also alleles. Alleles are the gene, but the nucleotide bases make up parts of the gene. So you have lots of different pairs of nucleotide bases, and then you have alleles that are also come in pairs on genes, one from your mom, one from your dad. So what I'm going to try to help you not get confused are the pairs of alleles versus the pairs of nucleotide bases. So we'll come back to that. Yeah, we did that. All right, so the DNA connection. So here's what I was just discussing. So transcription is the process by which the information in a strand of what is going to get copied. Do you know what we're going with here? So this is a strand of DNA, and it's going to get copied into a new molecule of messenger RNA, which we call mRNA. All right, DNA safely and stably stores genetic material in the nuclei of cells while as a reference or a template. Meanwhile, mRNA is comparable to a copy from a reference book because it carries the same information as DNA, but is not used for long-term storage and can freely exit the nucleus. Although the mRNA or messenger RNA contains the same information, it's not an identical copy because its sequence is complementary. So we're going to look at this a little more, but here's what I want you to notice. Do you notice there's a 5 and a 3 at the end? So what you call that is 5' and 3', which basically means the DNA has a direction. Like you have a head and you have feet, and this is kind of always what you think of as the top of your body and the bottom of your body. DNA has a leading end and a lagging end is what they call it. So when you're making new DNA, you need mRNA to come in and make like a temporary copy, a template that can be used to make new DNA. But if you notice the mRNA is pink right there, you see it? And you see that it has its 5 in line with the 3 from the DNA, and it has its 3 in line with the 5. Because you'll have a 5 going this way, okay? Like say this is 5, this is 3. So DNA is gonna lay like this. And when DNA unzips, the RNA that comes in is going to match up with the opposite primes. All right. So just kind of keep that in mind a little bit as we go on. So the translation is the process of translating the sequence of RNA or messenger RNA molecule to a sequence of what? What do you think we're talking about here? Anybody know? So it's amino acids. Now, last year, Evan, you weren't here, but last year at the end of the year, girls, do you remember the activity we did where you had a whole bunch of words and you had to make sentences? All right. So this is kind of how it works. So there's a code, and we work in 3s. So these are nucleotide bases where you see all those letters, T, A, C, G, G, C, G. See those? So those are nucleotide bases. And DNA has billions of those, but we read them in chunks of 3, CTA, TGC, whatever. And we read them in chunks of 3, and each chunk of 3 is going to get translated into a particular amino acid. It means, like GTC may mean make this particular amino acid, let's just call it amino acid A, all right? And then you get another combination, TCA, and that means make amino acid B. And the order in which the amino acids are put together determines what the protein is. And we need the protein because that's pretty much what makes up everything in the end. So a mutation, what's a mutation? So a mutation occurs when when a DNA gene is damaged or changed in such a way to alter the genetic message carried by the gene. So these are mutagens that cause mutations. So there are certain substances that you can become exposed to that can cause problems in your DNA. But first, let's look. Who sees the problem? Does anybody see the problem here? Where is it, Emerson? Where's the problem? Do you see it? The C. Yeah. Do you know what it look based on what you've seen in the past few slides? Do you know what? Well, look above. You see what that says up there? It says it's a T, and here it says it's a C. So in DNA, T and A always go together, and C and G always go together. So if this one changes, then if this DNA gets translated and mRNA comes in, it's going to make the opposite side of that pair different as well. So yeah, that C isn't supposed to be there. It's supposed to be a T. A lot of things can make that happen. Sometimes it's just misreading. Like when you're reading, do you ever read and just read the wrong word? Like you read the wrong word, or you're writing, say you're writing out a phone number and you write one digit wrong. You're supposed to write 678-403 and you write 678-503, right? So that sometimes it just happens. It just happens as part of the process. Other times there are things that you can be exposed to that can cause damage to your DNA. We call those mutagens, and some of them cause damage to your DNA in such a way that it causes your cells to not grow properly and to not stop growing. And do we remember what that's called? What is it when your cells don't stop growing? They can't stop, and they just grow and grow and grow and grow out of control. That's cancer, right? Very good. So there are some things you can take that will do that, like cigarette smoke. That's a good example, right? UV light. We talked about those things. All right, so here's how it works, what we were talking about. And you can see watching this, it's complicated. So you can see how mistakes could happen.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, A transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So you can see there's lots of different steps, and you can see that there's room for things to get, there's room for things to get messed up, right? There's room for mistakes to be made. And those are called mutations. Each of you probably has hundreds, thousands of mutations in your DNA. And a lot of them are not harmful at all. They won't cause you any problem. You've got enough redundancy, enough of the code in there that it's not going to cause an issue. But some of them, and we'll talk about those in a couple of weeks, some of them can be fatal. Some of them can cause big disabilities and things like that. So it depends on where that mutation is and what it causes. So when that mRNA comes in and is formed from the DNA, we call that transcription, because we're sitting there, we're basically making a copy, we're transcribing, like a person might transcribe. You can have in your cold case files, you have a transcriptions of your 911 call, you have transcriptions of all of the audio interviews, right, from all of the different witnesses that were interviewed, and that's a transcription. When the mRNA leaves the nucleus and goes into the cytoplasm, and then you start getting the little amino acids forming from the code in the mRNA, that's called translation, because now you're taking that information and like making it into a new language. So now it's no longer mRNA language, it's let's make a protein, and that protein is going to communicate in different ways in your body. Okay, so how is DNA discovered? Let's talk about this. So first we had this guy, Frederick Griffith, all right, and he worked with cute little mice and a pneumonia-causing bacteria. So a bacteria that causes pneumonia. Do you know what pneumonia is? Anybody know what pneumonia is? No? Nobody knows what pneumonia is? Pneumonia is a sickness in your lungs, and it often has to do with your lungs filling up with fluid, and it makes it very difficult to breathe when you have pneumonia. And it nowadays we can handle it most of the time, but it can be deadly if it's not treated. And way back before we had antibiotics and stuff, you could die from pneumonia. See, it says it's a bacteria, so you can probably treat it with an antibiotic, because bacteria can be killed by antibiotics, or it can impair the way they reproduce so that it doesn't cause any more problems. But viral infections, we don't have any, we have a couple meds you can take for them, but antibiotics don't work on viruses. That's because viruses aren't alive, but we'll talk about that later. So he concluded that genetic information could be transferred from one bacterial strain to another. So this is when one type of bacteria that is harmless is going to get changed permanently into another that causes diseases. And he called that transforming factor a gene. So now we have the term gene. Remember that Mendel talked about traits, right? He talked about traits, but he didn't really have that terminology. So now we have this terminology. And this is 1928. So almost 100 years ago, which probably seems like a really long time, but we know in the span of like scientific discovery, it's not all that long ago, right? And then next we get to the 1940s, and they call this Oswald Avery's experiment, but it was really three guys. It's Cullen McLeod, Macklin McCarty, and Oswald Avery. So we have some Scotsmen in here, right? So they discovered that DNA stores and transmits genetic information from one generation to the next. And they decided that DNA is responsible for that transformation. So they were able to look at the things that make up our cells, find a molecule in it that they think is responsible for transmitting that genetic information, those traits, right? So in the past, we understood, kids look like their parents, for example, or offspring look like their parents, but we couldn't really explain how that happened. So first they discovered there's something like genes, that's Griffith, right? There's some factor that is holding that information. And then we get to the point where, all right, we can pass that information from one generation to the next. And basically what he did is a whole series of steps where they had bacteria that they had killed some with heat and they had some that were living, and he put them with all of these different things to try and figure out what was carrying that information from one generation to the next. And he narrowed it down to DNA, basically by process of elimination. He eliminated lipids, he eliminated proteins, he eliminated sugars, he eliminated RNA. So the only thing left was DNA. That must be what's carrying the information. So then we get Hershey Chase. So this is Martha Chase and Alfred Hershey. And now we're in 1952. So studied probably around the time my mom had that haircut experience. So studied bacteriophage, composed of DNA core and a protein coat. Okay. Do you know what a bacteriophage is? This is crazy. A bacteriophage is a virus, but it only infects bacteria. And it looks like a flippin' spaceship. It really does. There's a picture, part of one right here. It looks almost like a lunar lander or something. It doesn't even look like it should be like a biological thing because it looks so engineered and mechanical. But it's this little thing, and it'll come along. It's a virus, and it'll land on a bacteria, and it'll inject its genetic material into the bacteria. So what they did is they wanted to determine what part was entering the bacterial cell. Was it injecting DNA into the cell, or was it injecting some kind of a protein coat that's around the little bacteriophage? And they were able to determine using radioactive markers that they were injecting their DNA. And then those host cells, the bacteria cells, would explode and release all of those new viruses, because a virus will infect a cell and cause that cell to reproduce its genetic material to make more of itself, right? So they were able to narrow it down and say, yep, all right. And then we get to 1949. Actually, those were out of order, weren't they? And we get Edwin Chargaff. Okay, so this guy is really important. He looked at the molecule of DNA, and he noticed that there are always equal amounts of A and T, the amounts of A and T, which are adenine and thymine. Remember we saw those ATGCs? The A and T of the amounts are always equal. And the G and C amounts are always equal. So what would that tell you? Think about it for a minute. If you didn't know anything about this molecule, but you knew that it contained adenine, thymine, cytosine, guanine, ATCG, and you saw that the amounts of A and T were always the same as each other, and the amounts of G and C were always the same as each other, what might that tell you about it? No? What do you think? What do you think? What do you think? What might you guess from that? They're always the same. The amount of A is always the amount is the same as T. And the amount of G is always the same as the amount of C. So what he thought is, okay, and this is very important to understand, because he was the first one to catch on to this. If a particular DNA molecule has the same amount of A as it does T, and the same amount of G as it does C, that must mean that A and T are paired. They go together. And G and C are paired. They go together. So that is very important, because this is our first understanding that when we start to go look into the structure of DNA, which is what we're trying to find out now, is how is this structured? That when we look into the structure of DNA, we're able to understand that that we're looking for pairs. So now is when we come to one of the most notorious scandals in science history. For those of you who did, let's see, you guys weren't with me last year. When we do physical science, we talk about, you were last year, but not year before last. We talk about Edison and Tesla. Edison and Tesla, there's a huge scandal there. It's a big, big scandal where like the two of them were basically at war with one another to see who would win, whose type of electricity would win to be used, and in what applications. And it went so far as electrocuting an elephant, literally. I'm not kidding. Electrocuting an elephant in front of a crowd of people. Yeah, it was a little while. So This one, though, has only recently been discovered. And by recently, I mean like only really recently admitted within probably the last 25-ish years that we really paid attention. So it starts with this young lady. Does anybody know who she is? Anybody know who she is? She's a little tiny person. It's hard to tell as a baby. So she was born on July 25th, 1920, and her name is Rosalind Franklin. She's British. She was born in London, okay? And as a young child, she was like really, really smart, and everybody noticed she was very smart. She attended a place called Norland Place School. And then eventually she went to St. Paul's Girls' School, which was one of the few places where girls could learn chemistry. Girls learned history, foreign language, art, English, literature. They didn't learn sciences, and they only learned basic math because that's not for girls. That's the way it used to be, right? So Rosalind really wanted to learn science stuff. So she went to St. Paul's Girls' School because there she could learn chemistry and physics, which they would not teach to girls in other schools. They wouldn't even teach it. And then she went on to this place called Newnham College. And in 1941, she graduated with what's called second class honors. What does that mean? Okay. Today, if you go to a 4-year college, do you know what they give you at the end of the 4 years if you earn it? What's it called? A degree. Do you know what it's called? A 4-year degree? It's called a bachelor's degree. And if you go 2 more, it's called a master's. If you run that, it's called like a doctoral degree, right? But your basic degree, if you go for 2 years, it's an associate's degree. But if you go for 4, which is your standard college, right, for 4 years, you earn a bachelor's degree. Women couldn't earn bachelor's degrees. Not a thing. Didn't even matter if you did all the work. You still didn't get the degree. Instead, they gave you what was called second class honors. Gee, second class is nice, right? So at the time, no bachelor's degrees for women. So she got herself this second class honors, which is as high as you can get. Good for her. She's doing her thing. So then she went on to Cambridge, which is a very well-known college, right? And she went and did a doctoral thesis, which means she is now going for a PhD or a doctoral degree. So she can be Dr. Franklin, right? Not a medical doctor, a doctor in this particular field. In this case, she's looking into chemistry, right? So she did her doctoral thesis, even without a bachelor's degree. They took her and said, come on, let's do this. And what she studied was called the physical chemistry of solid organic colloids with special reference to coal. She spent years looking at coal molecules under high-powered microscopes to try and determine the structure of coal. That's all it was. Holes in coal is what she called it. And she got her PhD in 1945. So then she asked a friend, keep an eye open for me. I'm looking for a job for someone who will hire a physical chemist who knows very little physical chemistry, but quite a lot about holes in coal. Very little is her term. By our standards, she knows a whole lot about physical chemistry, but she's thinking, I probably don't know all that much yet because I'm just graduating. I'm just getting out into the world. So what happened then is she got a job at King's College, which is in Cambridge, so nearby. So she gets this job, and here's what she did. She became an expert in what's called X-ray crystallography. And so it's a way to take an X-ray, but fire it through or at a lead screen. And then, so it blocks some of it, but it'll let a little ray go through. See how the one little ray goes through? Then it hits a little like prism and is diffracted. And then in there, in that little prismatic area is a molecule you're trying to look at. And it'll show a picture of the molecule. So she actually positioned just little molecular samples in these little blocks and shot x-rays at them to try and get pictures of a molecule. Can you imagine? Molecules are tiny, right? And she's getting pictures of them. So she worked in this field called X-ray crystallography and X-ray diffraction, seeking to produce photographic evidence of the physical structure of all kinds of different molecules. So in 1951, so she's been at her job a little while, she gets this student working for her called Raymond Gosling, all right? So his job was to sit there and take photo after photo after photo. Some of them turn out great, some of them are not so great. It's like, right? So she kept working and working, and he was taking photos, and he works for her. So technically, whatever he finds while he's working for her falls under her, like she's in charge, right? So he takes a photo called photo 51, right? So that's just a number. probably the number that day. And that year at a research presentation, Franklin described her findings. So photo 51 is actually a photo, the first photo of a DNA molecule. Remember, we don't know its structure. All we know is it has A and T in equal amounts, G and C in equal amounts. We know it has a phosphate sugar backbone. We know the components of DNA, but we don't know what it looks like. All right. So this is what it looks like. I'm wearing it. Did you notice I'm wearing it? So this is what it looks like. So what is this? This isn't, when you see DNA, you see the helix, right? This isn't looking at it sideways. This is looking down it. Like if this is your strand of DNA, it's looking at it like this, right down it, okay? So they're looking down into it like a cross section, and you see that X? That's where the twisting is going on. So that's what she got a picture of. So she said it suggests a helical structure which must be very closely packed, containing 2, 3, or 4 coaxial nucleic acid chains per helical unit, and having phosphate groups near the outside. All right, now, at the same time that she was taking pictures of DNA molecules to try and get a good enough picture to determine what it looked like, we had these two dudes. So in 1953, then she left, right, and went to a place called Birkbeck College. But as she was headed out the door, these guys had been working at the same time as her when she was taking all those photos, and their names are Francis Crick and James Watson. And they were trying to determine the structure of DNA too, but they weren't doing it using photos. They were using a model and trying to find out how it fit together because they knew the shape of the individual components. They know the shape of thymine, the shape of adenine, they know the shape of a phosphate, the shape of a sugar, and they were like trying to jam this puzzle together in a way that made sense. They weren't doing very well, but they were trying. They were coming along. So Watson and Crick had come close to creating a model that worked, but Franklin had been staunchly anti-model because she said that you should only create a model after you've confirmed the structure, rather than try and create a model and say, this makes sense, so it must be the structure. So here's the thing about her that you have to know. She was not well liked as a person. She was abrasive. She stared directly in your eyes when she talked to you, and people found that unnerving because she kind of stared you down. It was just sort of the way she was. She was kind of short with people, not warm and friendly. So people like that can be difficult to work with. But if we're being really honest, in the early 1950s and late 1940s, a woman whose behavior was like that, now she was really not liked because you're supposed to smile and be warm and be pretty. And she wasn't into that. That wasn't her jam. So James Watson, one of those two guys working on that model, he later wrote that he and his male colleagues called her Rosie. Okay, so when they talked about her, they called her Rosie. He and all the boys that he worked with, all the guys. And the name that people at King's College used behind her back. So that wasn't her name. Remember, her name is Rosalind, but they were calling her Rosie behind her back. And she didn't want to be called by that name because she had a great aunt named Rosie, and she didn't particularly like that great aunt, I don't think. And in the family, she was called Roz. So sometimes her family members would call her Roz instead of Rosalind, but nobody called her Rosie. And she didn't like that name, so they used it behind her back because they didn't like her. So it's kind of a way of being mean about her behind her back. So she made it clear to an American visiting friend whose name was Dorothea Racke, while sitting with her at Crick's table in the Eagle Pub. So sitting there with Crick and the crew, because they all, they were colleagues, even though they weren't really working together. Racke asked her how she wanted to be called, and she replied, I'm afraid it will have to be Rosalind. Most definitely not Rosie. Meaning like, stop calling me that behind my back. I don't like it, right? So then what happened is this is really contentious because even though these people are, contemporary and they're colleagues and they're working on the same project and they have the same goal in mind, they are competing for who's going to get it right. So there's a lot of competition. There's a lot of stress. Raymond Gosling showed photo 51 to this dude named Maurice Wilkins. All right. This guy is your spy in the story. And Maurice Wilkins takes the data, the photo and stuff, to Watson and Crick and goes, hey, you know what they got in Roslie's lab? Let me show you. Slide this photo over to you. So now Watson and Crick, who've been struggling to figure out the shape of DNA and the structure of it by using a model, have a photo to work from, right? Rosalind's photo. So Maurice, so Gosling showed it to Wilkins. Wilkins showed it to Watson and Crick. And now Watson and Crick were able to go, oh, this makes sense. Looking at this photo, we just need to change this. And now suddenly they have a functional model because they looked at Rosalind's photo. So upon seeing the model, Franklin was reported to have said, it's very pretty. How are they going to prove it? She can prove it because she has a photo. They can't prove it. They just think it works. The only reason they think it works is because they've seen her photo. So Watson and Crick submitted their research for publishing after Franklin submitted hers. Here's the important thing, okay? If you do research in science or medicine, it only becomes like official when it's published. And publishing is a big deal. There are certain like journals that publish research. So what you want is you want your research published in a journal. So she submitted hers for publishing. Right after she submitted hers, they submitted theirs for publishing. But here's where it all went crazy. The publication showed, thank you. It does that sometimes. So the publication where everybody submitted their research showed both. Oh, let me do the thing. Okay. It showed both Watson and Crick's research and Franklin's research, but they put Watson and Crick's before Franklin's. So when you're turning pages, you see Watson and Crick discovered DNA based on this model structure, and they toyed with the model until they got it right. And then you turn the page, and then you see Franklin got a picture of DNA. And people internalized that hers came after theirs, when in fact hers came before. Yes. So the credit for who found the structure of DNA for decades went to Watson and Crick, not Franklin. She did not get credit, even though she actually beat them to it. All right, so she died April 16th, 1958, at the age of 37. Do you want to guess what you might die so young from in this particular field of work? How was she taking those photos? What was she using? Do you remember what it was called? The process that she was using? Something crystallography. Do you remember? She was using X-ray crystallography. What do X-rays give you? Do you know?
What?
Well, they give you radiation, right? It's a certain type of radiation. Why when you go to the dentist, do they put that big lead apron on you? You know that big lead thing that it's really heavy, and then they take pictures of your teeth, but you're wearing this big heavy thing? Why do they do that? You know why they do that?
It's like an extra or something.
Yeah, they're trying to keep all that radiation out of the rest of your body, especially like your reproductive organs and whatever. That's why they cover you. They're just aiming at radiation just here. They don't want it to go everywhere. She had radiation every day. So what do you suppose happened to her?
Yeah.
Bingo. Cancer at the age of 37, dead. Ovarian cancer. So it got her reproductive system. So after all that work, she dies a few years later at the age of 37, probably from being exposed to radiation constantly in her work. And to top that off, as if that wasn't sad enough, she gets no credit for her discovery for decades. Now, if you look here at her grave, I want you to notice something. Do you see these, do you see the little figures right there above the rocks? Anybody see what that is?
Is that Hebrew?
That is Hebrew. Good, Evan. So what do you suppose that tells us about Rosalind Franklin? If there's Hebrew on her grave. She's Jewish. So that's another reason that it was hard for her. So she was a woman. She was not friendly. She was unnerving. She stared people down. She was abrasive, not warm and friendly. And she's Jewish. So these are all things that made it difficult for her to be liked and accepted and lauded. It just made her, it made the whole path to her success more difficult. And then despite her discoveries, she dies at 37 from cancer. And there was a Nobel Prize given for this discovery of the structure of DNA. And who got it? Watson and Crick got it. Now, here's something. You cannot get the Nobel Prize posthumously, which means after you die. If you're dead, you don't get a Nobel Prize. It doesn't matter what you discovered. You could discover the most amazing thing tomorrow, drop dead on Thursday, you're not getting a Nobel Prize. You have to be alive to receive it. So there's no chance that they could even go back and correct this and give her a Nobel Prize after she died and go, we're awarding this to her for her discovery. So it's done. History's done. That's it. They took the win. She took a hit and went on. So it went to Francis Harry Compton Crick, James Dewey Watson, and Maurice Hugh Frederick Wilkins, our spy. He even got the Nobel Prize for taking the picture from her and giving it to them, essentially, right? So, and their discoveries for concerning the molecular structure of nucleic acids and its significance for information transfer in living material. So, photo 51 was taken by Raymond Gosling, the graduate student working in Franklin's lab. The diffraction pattern determined the helical nature of the double helix strands, or that they were anti-parallel. And the outside of the DNA chain has a backbone of alternating deoxyribose and phosphate moieties is what they call them. And then the base pairs and the order which provide codes for the protein building, thereby the inheritance, are inside the helix. So that goes to Franklin, even though she didn't get credit till about 25 years ago. People started to be like, wait, you know what really happened though? And here's an interesting thing. You can actually pull up a TED Talk. If you like TED Talks, James Watson did a TED Talk about 15 years ago, and he still, in that TED Talk, said, there's been this whole issue that really it was Franklin, and I mean, she was working it, but we got it. We got it. We did it. Like, he still downplayed her role, even now, even just 15 years ago. So there's Maurice Wilkins, our spy. Here's Watson and Crick that used evidence from Franklin's photograph to determine their structure. And I'm going to come back to this one. All right. So I'm going to make sure you got this. So here's your information for these. If you want to check anything in your binder, we'll leave that up for a second. And let me go back now and we'll do that video. I just want to make sure you have a chance to do that. And then if you have any time afterwards, you can work on dragons. You might have you probably won't, but let's see. Oh, it's commercial. There you go.
The discovery of the structure of DNA was one of the most important scientific achievements in the last century, in human history, in fact. The now famous double helix is almost synonymous with Watson and Crick, two of the scientists who won the Nobel Prize for figuring it out. But there's another name you may know too, Rosalind Franklin. You may have heard that her data supported Watson and Crick's brilliant idea, or that she was a plain-dressing, belligerent scientist, which is how Watson actually described her in The Double Helix. But thanks to Franklin's biographers, who investigated her life and interviewed many people close to her, we now know that that account is far from true, and her scientific contributions have been vastly underplayed. Let's hear the real story. Rosalind Elsie Franklin was born in London in 1920. She wanted to be a scientist ever since she was a teenager, which wasn't a common or easy career path for girls at that time. But she excelled at science anyway. She won a scholarship to Cambridge to study chemistry, where she earned her PhD. And she later conducted research on the structure of coal that led to better gas masks for the British during World War II. In 1951, she joined King's College to use X-ray techniques to study the structure of DNA, then one of the hottest topics in science. Franklin upgraded the X-ray lab and got to work, shining high-energy X-rays on tiny wet crystals of DNA. But the academic culture at the time wasn't very friendly to women, and Franklin was isolated from her colleagues. She clashed with Maurice Wilkins, a lab mate who assumed Franklin had been hired as his assistant. But Franklin kept working, and in 1952, she obtained Photo 51, the most famous X-ray image of DNA. Just getting the image took 100 hours. The calculations necessary to analyze it would take a year. Meanwhile,
The American biologist James Watson and the British physicist Francis Crick were also working on finding DNA structure. Without Franklin's knowledge, Wilkins took photo 51 and showed it to Watson and Crick. Instead of calculating the exact position of every atom, they did a quick analysis of Franklin's data and used that to build a few potential structures. Eventually, they arrived at the right one. DNA is made of two helicoidal strands, one opposite the other, with bases at the center like rungs of a ladder. Watson and Crick published their model in April 1953. Meanwhile, Franklin had finished her calculations, come to the same conclusion, and submitted her own manuscript. The journal published the manuscripts together, but put Franklin's last, making it look like her experiments just confirmed Watson and Crick's breakthrough, instead of inspiring it. But Franklin had already stopped working on DNA and died of cancer in 1958, never knowing that Watson and Crick had seen her photographs. Watson, Crick, and Wilkins won the Nobel Prize in 1962 for their work on DNA. It's often said that Franklin would have been recognized by a Nobel Prize if only they could be awarded posthumously. And in fact, it's possible she could have won twice. Her work on the structure of viruses led to a Nobel for a colleague in 1982. It's time to tell the story of a brave woman who fought sexism in science and whose work revolutionized medicine, biology, and agriculture. It's time to honor Rosalind L.C. Franklin, the unsung mother of the double helix.
Good old Rosalind Franklin. All right, so grab your pencils, grab your lab, and let's get ready.